Hodgkin and Huxley conducted voltage clamp experiments on the squid giant axon to evaluate the ionic basis of the action potential. These were undoubtedly some of the most important experiments ever conducted in neuroscience. They revealed to us how the action potential works in all animals, and the action potential, as we shall see, is the universal language of the nervous system. In this movie, I will first introduce you to the squid giant axon, and then show what Hodgkin and Huxley found in their initial studies. I will then explain the reasons why they had to use voltage clamp to answer the questions they were asking. In the second movie, I will show how voltage clamp actually works, and how Hodgkin and Huxley used the voltage clamp technique to reveal the sodium and potassium currents that generate the action potential. This is the animal Hodgkin and Huxley used in their experiments, the squid, calamari. They used it because the squid has giant axons running down the side of its body. The axons are so huge they have very, very high conduction velocities, and they're used for escape. Indeed, they're about a millimeter in diameter and several centimeters long. Scientists can cut the axons out of the squid. They can put them in a dish. They can squeeze the axoplasm out of the axon. They can fill it with anything they want to. They can poke electrodes into the axon. They can push electrodes down the axon. And they can conduct experiments that would be impossible to conduct with smaller cells. To give you some appreciation for the dimensions and the elegance of this axon, the next movie shows a very brief presentation of how the squid giant axon is prepared for an experiment.
It usually takes about half an hour to clean an axon and to leave it bare of all the small fibers, often over a distance of about 5 centimeters. When finished, the giant axon alone remains, a single animal cell several centimeters in length and, in this case, about 650 microns in diameter.
Before I explain the mechanics of voltage clamping, let me explain the nature of the problems Hodgkin and Huxley encountered and the questions that they wanted to answer. On the left side is a drawing of a squid giant axon placed in a dish and immersed in normal seawater. One of the first things that Hodgkin and Huxley did was to impale the axon with a microelectrode. And when they did, they saw the resting potential. In fact, it was the first time that anyone had ever recorded a resting potential from a living cell. And they saw that the resting potential was close to the potassium equilibrium potential, and they knew that there had to be a potassium conductance in the cell. The next thing they did was to evoke an action potential, and they saw immediately that the peak of the action potential went far beyond zero. It went to about plus 55 millivolts. From this, they knew that there had to be a sodium conductance as well. There had to be a flow of sodium across the membrane. So they knew from these two things that the axon had to have both a sodium and a potassium conductance. To make the case for sodium conductance even stronger, they next changed the seawater bathing the axon and filled the dish with a solution that had a sodium concentration lower than the normal seawater. In other words, they lowered the external concentration of sodium. Now, when they evoked an action potential, they found that the peak of the action potential was also reduced, and the reduction was proportional to the reduction in the external concentration of sodium. From this, they knew that the peak of the action potential had to be dependent on some kind of sodium conductance. What they wanted to know is exactly how do the sodium and potassium currents produce an action potential. But they also had a problem. The problem is that the action potential varies in time. Consider the electrode on the left that impales the axon at one point along its membrane. When an action potential occurs at that point, the membrane potential changes constantly in time. Indeed, it is the change in membrane potential over time that defines the action potential. To make matters worse, the space over which the membrane potential changes also varies. When an action potential first occurs in the far left portion of the axon, it travels down the axon. So the membrane potential is changing not only in time, but in space as well. How does one measure a separate sodium and potassium current when the membrane potential is continuously changing in both time and space? As sodium and potassium currents change, so does the membrane potential, and once the membrane potential changes, the currents also change. In these circumstances, there is no way to disambiguate how the changes in membrane potential drive the currents, and how the changes in currents drive the membrane potential. So to cope with these problems, Hodgkin and Huxley had to do two things. First, they had to somehow clamp the membrane potential so that it would not change over time, and then measure the currents that were flowing into and out of the cell at that fixed membrane potential. And second, they had to clamp the membrane potential along the entire axon simultaneously so that the membrane potential did not vary in different places along the axon. But that's what voltage clamp does. It clamps the entire axon and holds it at a fixed voltage or membrane potential. In the next movie, we'll see how the voltage clamp system actually works.